So we are discussing about gametogenesis, oogenesis and spermatogenesis. So gametogonia are usually seen as the initial stage of gametogenesis. However, gametogonia are themselves successors of primordial germ cells or PGCs from the dorsal endoderm of the yolk sac migrate along the hindgut to the gonadal ridge. So they multiply by mitosis, and once they have reached the gonadal ridge in the late embryonic stage, are referred to as gametogonia. Once the sperm cells have developed into gametogonia, they are no longer the same between males and females. Individual path from gametogonia, males and female gametes develop differently, males by spermatogenesis and females by oogenesis. However, by convention, the following pattern is common for both cell types. Ploidy, both are diploid 2n46 condition. DNA copy 2C before replication, 4C after 46 before 26 into 2 after. Then processes gametogenesis, mitosis in case of gametogonia. Then primary gametocyte turn into gametidogenesis, meiosis 1. And secondary gametocyte, gametidiogenesis. Then gametid, haploid. Primary oocyte, secondary oocyte, mature ovum, polar bodies. Primary spermatocyte, secondary spermatocytes, spermatids. Spermatogenesis. Spermatogenesis is the process by which haploid spermatozoa develop from germ cells in the seminiferous tubules of the testis. The entire process of spermatogenesis can be broken up into several distinct stages, each corresponding to a particular type of cell in humans. In the following table, ploidy, copy number and chromosome chromatid counts are for one cell genetically prior to DNA synthesis and division in G1 if applicable. The primary spermatocyte is arrested after DNA synthesis and prior to division. In spermatocytogenesis. Spermatocytogenesis is the male form of gametocytogenesis and results in the formation of spermatocytes possessing half the normal complement of genetic material. In spermatocytogenesis, a diploid spermatogonium, which resides in the basal compartment of the seminiferous tubule, divide mitotically, producing two diploid intermediate cells called primary spermatocytes. Each primary spermatocyte then moves into the adluvial compartment of the seminiferous tubules and duplicate its DNA and subsequently undergoes meiosis 1 to produce two haploid secondary spermatocytes, which will later divide once more into haploid spermatids. This division implicates sources of genetic variation such as random inclusion of either parental chromosomes and chromosomal crossover that increase the genetic variability of the gamete. The DNA damage response (DDR) machinery plays an important role in spermatogenesis. The protein FMRP binds to meiotic chromosomes and regulates the dynamics of the DDR machinery during spermatogenesis. So FMFR appears to be necessary for the repair of DNA damage. Each cell division from a spermatogonium to a spermatid is incomplete. The cells remain connected to one another by bridges of cytoplasm to allow synchronous development. Not all spermatogonia divide to produce spermatocytes, otherwise the supply of spermatogonia would run out. Instead, spermatogonial stem cells divide mitotically to produce copies of themselves, ensuring a constant supply of spermatogonia to fuel spermatogenesis. Process of spermatogenesis as the cell progresses from primary spermatocytes to secondary spermatocytes to spermatids to spermatidogenesis. Spermatidogenesis is the creation of spermatids from secondary spermatocytes. Secondary spermatocytes produce earlier rapidly enter meiosis 2 and divide to produce haploid spermatids. The brevity of this stage means that secondary spermatocytes are rarely seen in histological studies. Then spermiogenesis. During spermiogenesis, the spermatids begin to form a tail by growing microtubules on one of the centrioles, which turns into basal body. Thus, microtubules form an axone. Later, the centriole is modified in the process of centrosome reduction. The anterior part of the tail, called the midpiece, thickens because mitochondria are arranged around the axone to ensure energy supply. Then spermatid DNA also undergoes packaging, become highly condensed. The DNA is packaged firstly with specific nuclear basic proteins, which are subsequently replaced with protamins during spermatid elongation. The resultant tightly packed chromatin is transcriptionally inactive. The Golgi apparatus surrounds the new condensed nucleus, becoming the acrosome. So maturation then takes place under the influence of testosterone, which removes the remaining unnecessary cytoplasm and organelles. The excess cytoplasm, known as residual bodies, is phagocytosed by surrounding Sertoli cells in the testis. The resulting spermatozoa are now mature but lack motility. The mature spermatozoa released from the protective Sertoli cells into the lumen of the seminiferous tubule in a process called spermiation. The non-motile spermatozoa are transported to the epididymis and testicular fluid secreted by the Sertoli cells, which with the aid of peristaltic contraction. While in the epididymis, the spermatozoa gain motility and become capable of fertilization. The spermatozoa gain motility and become capable of fertilization. However, transport of the mature spermatozoa through the remainder of the male reproductive system is achieved via muscle contraction rather than the spermatozoan's recently acquired motility. Then role of Sertoli cells. At all stages of differentiation, the spermatogenic cells are in close contact with Sertoli cells, which are thought to provide structural and metabolic support to the developing sperm cells. A single Sertoli cell extends from the basement membrane to the lumen of the seminiferous tubule. Although the cytoplasmic processes are difficult to distinguish at the light microscopic effect, Sertoli cells serve a number of functions during spermatogenesis. They support the developing gametes in the following ways: maintain the environment necessary for development and maturation via the blood-testis barrier, secrete substances initiating meiosis, secrete supporting testicular fluid, secrete androgen secreting protein ABP, which concentrates testosterone in close proximity to the developing gametes. So testosterone is needed in very high quantities for maintenance of the reproductive tract, and ABP allows a much higher level of fertility. So secrete hormones affecting pituitary gland control of spermatogenesis, particularly the polypeptide hormone, inhibit. So phagocytose residual cytoplasm left over from spermiogenesis. Secretion of anti-Mullerian hormone causes deterioration of the Mullerian duct, then protect spermatids from the immune system of the male via the blood-testis barrier, contribute to the spermatogonial stem cell niche. The intracellular adhesion molecule ICAM1 and soluble ICAM1 have antagonistic effects on the tight junctions forming the blood-testis barrier. ICAM2 molecules regulate spermatid adhesion on the apical side of the barrier towards the lumen. Then oogenesis, or in case of human, oogenesis is the differentiation of the ovum or egg cell into the cell competent to further development. When fertilized, it is developed from the primary oocyte to the maturation. Oogenesis is initiated in the embryonic stage. In humans, oogenesis starts with the process of developing primary oocytes, which occur via the transformation of oogonia into primary oocytes, a process called oocytogenesis. So, oocytogenesis is complete either before or shortly after birth. Number of primary oocytes. It is commonly believed that when oocytogenesis is complete, no additional primary oocytes is created, in contrast to the male process of spermatogenesis, where gametocytes are continuously created. In other words, primary oocytes reach their maximum development at 20 weeks of gestational age, and approximately 7 million primary oocytes have been created. However, at birth, this number has already been reduced to approximately 1 to 2 million. Then ootidogenesis. Ootidogenesis, in the sense, the surrounding phase of ootidogenesis occurs when the primary oocyte develops into an ootid. This is achieved by the process of meiosis. In fact, a primary oocyte is, by its biological definition, a cell whose primary function is to divide by the process of meiosis. However, although this process begins at prenatal age, it stops at prophase 1. So there will be a start page at prophase 1. In late fetal life, all oocytes, still primary oocytes, have halted at this stage of development called the dicterate. After menarche, these cells then continue to develop, although only a few do so by every menstrual cycle. So, oogenesis in eukaryotic cells, oogonium, where the mitotic division occurs, differentiation and meiosis begins. Primary oocyte, meiosis 1 is completed and meiosis 2 begins. Secondary oocyte, first polar body, ovulation must occur, and the presence of the sperm penetration or fertilization induces meiosis 2 to completion. Then ovum, second polar body. So, what is happening in meiosis 1? Meiosis 1 of ootidogenesis begins during embryonic development but halts in the diplotin stage of prophase 1 until puberty. The mouse oocyte in the dictate or prolonged diplotin stage actively repairs DNA damage, whereas DNA repair is not detectable in the predictate leptotin, zygotin, and pachytene stages of meiosis. For those primary oocytes that continue to develop in each menstrual cycle, however, synapses occurs and tetrads form, enabling chromosomal crossover to occur. As a result of meiosis 1, the primary oocyte has now developed into the secondary oocyte and the first polar body. Then meiosis 2, immediately after meiosis 1, the haploid secondary oocyte initiates meiosis 2. However, this process is also halted at the metaphase 1 stage until fertilization, if such should ever occur. If the cell egg is not fertilized, it is disintegrated and released. That means menstruation, and the secondary oocyte does not complete meiosis 2 and doesn't become an ovum. When meiosis 2 has completed and ooted and other polar body have now been created, the polar body is small in size. Then folliculogenesis. Folliculogenesis, synchronously with ootidogenesis, the ovarian follicle surrounding the ootid has developed from a primordial follicle to a preovulary one. So, maturation into ovum. When both polar bodies disintegrate at the end of meiosis 2, leaving only the ootid, which then eventually undergoes maturation into a mature ovum. The function of forming polar bodies is to discard the extra haploid sets of chromosomes that have resulted as a consequence of meiosis. Then oogenesis in non-animals. In Ascaris, the oocyte does not even begin meiosis until the sperm touches it, in contrast to mammals, where meiosis is completed in the yeast cycle. In female Drosophila flies, genetic recombination occurs during meiosis. This recombination is associated with formation of DNA double-strand breaks and the repair of these breaks. The repair process leads to crossover recombinants as well as at least 3 times as many non-crossover recombinants arising by gene conversion without crossover. Now we are going to learn some aspects related to the ultrastructure of gametes. First, we are going to learn about sperm ultrastructure. Stanton van Leeuwenhoek, the Dutch microscopist who co-discovered sperm in 1678, first discovered them to be parasitic animals living within the semen, hence the term spermatozoa, meaning sperm animals. So, Nicholas Hudsocker, the other co-discoverer of sperm, drew a picture of what he hoped to find a preformed human or homunculus within the human sperm. So, this belief that the sperm contained the entire embryonic organism never gained much acceptance, as it implied an enormous waste of potential life. The first evidence suggesting the importance of sperm and reproduction came from a series of experiments performed by Lazzaro Spallanzani in the late 1970s. Spallanzani demonstrated that filtered toad semen devoid of sperm would not fertilize eggs. He concluded, however, that the viscous fluid retained by the filter paper and not the sperm was the agent for fertilization. He, like many others, felt that the spermatic animals were parasites. So each sperm consists of a haploid nucleus, a propulsion system to move the nucleus, and a sac of enzymes that enable the nucleus to enter the cell. Most of the sperm cytoplasm is eliminated during maturation, leaving only certain organelles that are modified for spermatic function. During the course of sperm maturation, the haploid nucleus becomes very streamlined and its DNA becomes tightly compressed. In front of its compressed haploid nucleus lies the acrosomal vesicle or acrosome, which is derived from the Golgi apparatus and contains enzymes that digest proteins and complex sugars. Thus, it can be considered a modified secretory vesicle. These stored enzymes are used to lyse the outer covering of the egg. In many species, such as sea urchins, a region of globular actin molecules lies between the nucleus and the acrosomal vesicle. These proteins are used to extend a finger-like acrosomal process from the sperm during the early stages of fertilization. In sea urchins and several other species, recognition between sperm and egg involve molecules on the acrosomal processes. So, together, the acrosome and nucleus constitute the head of the sperm. So, we were learning about that. The means by which sperm are propelled vary according to how the species has adapted to environmental conditions. In some species, such as parasitic roundworm Ascaris, the sperm travel by the amoeboid motion of lamellipodial extensions of the cell membrane. And in most species, however, each sperm is able to travel long distances by whipping its flagella. Flagella are complex structures. The major motor portion of the flagellum is called the axon. It is formed by microtubules emanating from the centriole at the base of the sperm nucleus. The core of the axon consists of two central microtubules surrounded by a row of 9 doublets microtubules. Actually, only one microtubule of each doublet is complete, having 13 protofilaments. The other is C-shaped and has only 11 protofilaments. Although tubulin is the basis for the structure of the flagellum, other proteins are also critical for flagella function. The force of sperm propulsion is provided by dynein, a protein that is attached to the microtubules. Dynein hydrolyzes molecules of ATP and can convert the released chemical energy into the mechanical energy that propels the sperm. Then ovum. All the material necessary for the beginning of growth and development must be stored in the mature egg or ovum. Whereas the sperm has eliminated most of its cytoplasm, the developing egg called the oocyte before it reaches the stage of meiosis at which it is fertilized, not by conserves its material, but is actively involved in accumulating more. The meiotic division that form the oocyte conserves its cytoplasm rather than giving half of it away. And the oocyte either synthesizes or absorbs proteins such as yolk that act as food reservoirs for the developing embryo. These birds and eggs are enormous single cells swollen with their accumulated yolk. Even eggs with relatively sparse yolk are comparatively large. The volume of sea urchin egg is about 200 picoliters. So, while sperm and egg have equal haploid nuclear components, the egg also has a remarkable cytoplasmic storehouse that it has accumulated during its maturation. The cytoplasmic trove include the following: vitelline envelope, jelly layer, sperms are there, plasma membrane, cortical granule, mitochondria, then nucleus. Proteins. It will be a long while since the embryo is able to feed itself or obtain food from its mother. The early embryonic cells need a supply of energy and amino acids. In many species, it is accompanied by accumulating yolk proteins in the egg. Many of the yolk proteins are made in other organs, liver, fat body, and travel through the maternal blood to the egg. Then ribosomes and tRNA, the early embryo needs to make many of its own proteins, and in some species there is a burst of protein synthesis soon after fertilization. So protein synthesis is accomplished by ribosomes and tRNA which exist in the egg. The developing egg has special mechanisms to synthesize ribosomes, and certain amphibian oocytes produce as many as 1.12 ribosomes during their meiotic prophase. Then messenger RNA. In most organisms, the instructions for proteins made during early development are already packaged in the oocyte. It is estimated that eggs of sea urchins contain 25,000 to 50,000 different types of mRNA. This mRNA, however, remains dormant until after fertilization. Then morphogenic factors. Molecules that direct the differentiation of cells into certain cell types are present in the egg. They appear to be localized in different regions of the egg and become segregated into different cells during cleavage. Then protective chemicals are there. The embryo cannot run away from the predators or move to a safe environment. So it must come up equipped to deal with threats. Many eggs contain ultraviolet filters and DNA repair enzymes that protect them from sunlight. Some eggs contain chemicals or molecules that potential predators find distasteful. And the yolk of the bird eggs even contain antibodies. Within this enormous volume of cytoplasm resides a large nucleus. Then in some species, like sea urchins, the nucleus is already haploid at the time of fertilization. In other species, including many worms and most mammals, the egg nucleus is still diploid and the sperm enters before the meiotic divisions are completed. So enclosing the cytoplasm is the egg plasma membrane. The membrane must regulate the flow of certain ions during fertilization and must be capable of focusing with the sperm plasma membrane. Outside the plasma membrane is the vitelline envelope, which forms a fluid fibrous mat around the egg. This envelope contains at least 8 different glycoproteins and is often involved in sperm-egg recognition. It is supplemented by extensions of membrane glycoproteins from the plasma membrane and by proteinaceous vitelline posts that adhere the vitelline envelope to the membrane. The vitelline envelope is essential for the species-specific binding of sperm. In mammals, the vitelline envelope is a separate and thick extracellular matrix called the zona pellucida. The mammalian egg is also surrounded by a layer of cells called the cumulus, which is made up of ovarian follicular cells that were nurturing the egg at the time of its release from the ovary. So mammalian sperm have to get past the cells to fertilize the egg. The innermost layer of cumulus cells immediately adjacent to the zona pellucida is carried to the corona radiata. Laying immediately beneath the plasma membrane of the egg is a thin shell about 5 mm of gel-like cytoplasm called the cortex. The cytoplasm of this region is stiffer than the internal cytoplasm and contains high concentrations of globular actin molecules. During fertilization, these actin molecules polymerize to form long cables of actin known as microfilaments. So microfilaments are necessary for cell division and they also are used to extend the egg surface into small projections called the microvilli, which may aid sperm entry into the cell. Also within the cortex are the cortical granules. These membrane-bound structures, which are homologous to the acrosomal vesicle of the sperm, are Golgi-derived organelles containing proteolytic enzymes. However, whereas each sperm contains one acrosomal vesicle, such each sea urchin egg contains approximately 15,000 cortical granules. Moreover, in addition to digestive enzymes, the cortical granules contain mucopolysaccharides, adhesive glycoproteins, and hyaline proteins. The enzymes and mucopolysaccharides are active in preventing other sperm from entering egg after the first sperm has entered, and the hyaline and adhesive glycoproteins surround the early embryo and provide support for the cleavage state blastomeres. Many types of cells also have an egg jelly outside the vitelline envelope. This glycoprotein meshwork can have numerous functions, but most commonly is used either to attract or to activate sperm. The egg then is a cell specialized for receiving sperm and initiating development. So cell surface molecules in sperm egg recognition in animals. The interaction of sperm and egg generally proceeds according to 5 basic steps. The chemoattraction of the sperm to the egg by soluble molecules secreted by the egg. The exocytosis of the acrosomal vesicle to release its enzymes. The binding of the sperm to the extracellular envelope vitelline layer of zona pellucida of the egg. The passing of the sperm through its extracellular envelope. Then fusion of egg and sperm cell plasma membrane. The chemoattraction of the sperm to the egg by soluble molecules secreted by the egg. The exocytosis of the acrosomal vesicle to release its enzymes. The binding of the sperm to the extracellular envelope or vitelline layer of zona pellucida of the egg. The passing of the sperm through the extracellular envelope. Fusion of egg and sperm cell plasma membranes.